Hello, BNG 426 class. This is the pre-lecture for tomorrow's flipped class, the Metabolic Network class. So we're going to be talking about construction of networks, quote-unquote, from scratch. Basically from the beginning is what I mean by that. There's no real making them from scratch, obviously, but we can begin to think about them from scratch. As it were. So we take an interest in a gene, an enzyme, a product, a pathway as metabolic engineers or as people that manipulate organisms in order to make some sort of product. Our aim to study, improve the pathway. We subject that organism that has that pathway to perturbations, and these perturbations affect more than just the gene and pathway of interest. In fact, they probably do a lot of things that we don't really know about, at least on the surface, because we can't see what's going on in the cell. Although they might do also some things that you don't know about initially, but then find out because they don't grow well, or they grow slower or faster, or something. So perturbations can have a lot of unforeseen consequences. And part, I think, of metabolic networks is, studying metabolic networks anyway, is to try to get in front of as many unforeseen consequences as you possibly can in terms of metabolic engineering. So metabolic networks can be examined to tell us about how what we are doing might affect other pathways. And again, getting in front of those unforeseen consequences. And it also tells us about what we might be doing that affects the cell. Because basically perturbing any pathway is probably going to have some effect on the cell. And maybe we can't see that initially. So we could be affecting energetics, cell division, storage of reducing power, stress response, things like that. We want to study these things beyond just the pathway level. It's well and good to take a pathway and make some changes to it, do some tinkering, and say, hey, we've made more product, but we need to go beyond the pathway level sometimes. So here is a good example. You can see how oral bacteria respond, and this is a metabolic network of oral bacteria, how they respond to you being sick, you the host being sick. And as you can plainly see, there are a couple of pathways, there are at least 4 pathways here in yellow that are involved in how they respond. So simply just going by the pathway route is not going to cover it all. We're going to need to do more of a metabolic network and find out how these 4 pathways, or these 5 pathways, are linked. Find out how they are linked. And metabolic networks and network flux analysis is one way in which we can do that. It's a good way for us to put the pieces together to make one big puzzle here. But how do we get from just a bunch of pathways to a network? How does a network first come into being, at least in terms of as a thing that we study? So in terms of experiments, what is needed, we need to know what is needed to get enough data to begin constructing a network. Is just having a whole bunch of DNA sequences, is that enough? Or do we need to have more in hand before we get started? So every network would start with a genome. It's kind of hard to construct a network if you don't know what genes your organism has and how they fit together, at least in terms of what's going on with the DNA message. Genome data, of course, is relatively easy to get these days. It might take a couple of weeks to a couple of months to sequence a genome and to annotate it using tools that are available on the NCBI and to get some annotated genome data that can be the basis, the foundation of a network. You will want additional reinforcing data for your network, things like gene expression data, transcriptomics, to tell you what genes are being expressed to what levels under what conditions. And of course, the genes, the levels, the conditions, that all depends on the thing you're studying, the phenomenon that you're studying. In addition, you can use things like proteomics. It tells you how much protein or how much enzyme is available. Metabolomics, it tells you how much product is being made, how many intermediates are being made, how many intermediates are in the cell. And you can also use comparative genomics, which can be, at least on the face of it, it can be a DNA to DNA comparison of different strains and species, or at least the genomes of those strains and the genomes of those species. So we showed this figure on Monday, but it is relevant here because we are testing a network. We have basically built the foundation and we want to test it. So we want to see if we can fill in the knowledge gaps, as part A said. But certainly methods, not only method A, but methods A, B, and F can certainly provide refinement of a metabolic network. And depending on what you're doing, methods C, D, and certainly method E would be of help as well. And certainly E is good if you want to get physiological data that helps you refine your network of choice. Okay, so let's do a crude, unnamed example here. Let's look at some metabolites. So we have random assortment of metabolites here. We'll just call them number 1 through number 9. We know that we care about some of them. One of them might be our product. One of them might be the substrate that gets us to our product. But how do the others relate? What we essentially want to know is how many degrees of separation are there between metabolites. Is number 9 directly involved with number 8? Is it peripherally involved with number 3? We need to know this stuff in order to generate our network. Okay, so we have number 2 and number 9 are our metabolites that we're interested in. Number 9, let's say, is our substrate, and number 2 is our product. And there's a pathway in these 9 metabolites somewhere. Likely, at least, some of these metabolites will be closely involved in making number 2 from number 9, and that means that those metabolites are intermediary metabolites that are part of the pathway. So that can be pretty much where we would want to start us, to at least define the pathway. So initially we might want to do some stoichiometry. If we think that number 9 is a 6-carbon compound and number 2 is a 3-carbon compound, we might know that every mole of number 9, 2 moles of number 2 are produced. So then we might want to indicate that on this little grid here. So we read 2-2, which would be 2 moles of number 2 for every 1 mole of number 9 on the grid here. And this is important if we are examining control of flux, how much carbon is flowing through this pathway, and ultimately through the network that we might create with these metabolites, including and not limited to the pathway. So the first step, and I think this gets us often, if not always gets us the pathway that we are most interested in, is we look at genomics. We take the genome of the organism, we use whatever our favorite database is, and we look at the annotations of the genes that potentially could be part of the pathway we're interested in. So the gene annotations will tell us that certain genes might have certain names, and in this case the annotations would give us an idea of what the pathway looks like. So in this case we see the pathway starts at number 9 as the substrate, number 8 is an intermediate, then number 5, then clearly number 5, still a 6-carbon compound, splits to 2 number 2s. And those are our products there. Okay, so a little bit more about gene annotation. The annotation of genes is often about educated guesses. Annotation of genes can be like a good mystery novel or a crime scene investigation show. The more evidence you have, the more likely you will be getting your predictions correct. The more likely you will figure it out. So gene annotations, in the absence of physiological and or biochemical data, are merely predictions. And that's not to downplay these annotations or downplay predictions in general. It's just that physiological data and biochemical data could pretty much confirm that your annotation is correct, or it could disprove an annotation that is incorrect. So those help build evidence in your favor or potentially against you if you've made an incorrect annotation. So for our pathway here, or for our network that we want to make, we might want to ask other questions like, are parts of this pathway reversible? Are there any carbon sinks? We have product 2 here. Are there any sinks like product 4 or 7 or whatever that are taking carbon away from product 2? We would want to know that. Are there other ways to make the product? Does number 3, metabolite 3, make number 2? We don't know that yet. And how does the key pathway of interest fit in with cellular metabolism? We probably can't ask that question here unless the cell that this pathway is a part of only makes 9 metabolites, which is very unlikely. But anyway, that is a question that could certainly be asked in a bigger system. So we can use biochemical data because that can tell us whether a reaction is reversible or not. One of the big caveats to this, however, is in vitro enzymology. So we take enzyme, put it in a tube with substrate and whatever cofactors and buffer. It's not the same as an in vivo functioning pathway. It's not necessarily indicative of what goes on in the cell because concentrations are different. Conditions, whether you like it or not, are going to be different, at least a little bit. So it might not necessarily reflect real life. Ideally, what you would want to use is the combination of biochemical and metabolic/physiological data to confirm, at least in directionality of a reaction in a pathway, to confirm this. So, for example, we can say that we have both of those things because we're just making this up. Biochemical and physiological data will confirm that the reaction 9-8 and the reaction 8-5 are reversible, so they go both ways here. We can have 8, depending on the concentration of 8, we can have 8 making 5, or if there's too much 5, 5 might go back and make some 8, depending what's going on. We can also use transcriptomic data, and that can be very helpful. We can count transcripts, RNA, to get an idea of gene expression. And if we can find that high expression of genes under certain conditions, conditions that we care about, that can suggest or confirm roles in a network. So if under conditions favorable for production of metabolite 2, the enzyme responsible for making number 5 from number 7 is upregulated, we might know already that number 7 can be converted to number 5 by one enzyme, by one reaction. But we have shown, looking at either a microarray or RNA-seq or what have you, we have shown that indeed under conditions favorable for making 2 from 9, that number 7 is also adding to this by making number 5 and probably ultimately making number 2. Then maybe that reaction should be part of our network here. And of course we can also use experimental data. Sometimes there's just no substitute for experimentation. It'll tell you, we can find out, oh, something else is being made. We're getting byproducts made here. So if we look at culture broths, we can see, okay, we have number 2, our metabolite number 2, but look, we're also getting a lot of number 1. I wonder why that is. We do a little bit more hunting, do some metabolomics, and we can find that number 1 is being made under these conditions from number 4 as its precursor. And this is under conditions, again, conditions favorable for production of number 2. It doesn't necessarily mean much on its own because you can see maybe it's part of the pathway, but it's not connected to the main pathway. Certainly it is part of the network, but what we can do is then take that a little bit further. Say we do some radio-labeled substrate experiments, either add radio-labeled number 9 or radio-labeled number 7 or a combination thereof, and show that at least a fraction of number 1 is being made using number 5 as a precursor. So we need to consider number 5 to number 4, that reaction, and number 4 to number 1, that reaction, as part of our metabolic network that we're going to look at. So we could also compare the genes that may or may not be in our network to other organisms. How do other closely related organisms do their business? And that can help uncover how our organism does its thing. If strain B of an organism has a recycle pathway that feeds off of and into our pathway of choice, and perhaps our organism can do that too. So we can start by comparing genomic data of two strains. We can also do physiological comparisons. We can do metabolomic comparisons here, and we can find out that, hey, what do you know? We have number 8 making number 6, which makes number 3, and then number 3 is making number 8 again, which on the face of it looks like a feudal cycle. Maybe it isn't. We're not going to take this any further, though, but just to say that we can compare and see that one strain is doing it. We can find out, using what is known about that strain, we can apply that to our strain and show that, well, it's happening here too. So there are many ways to unearth a comprehensive metabolic network. Different experiments, and we've talked about a few of the different experiments that we could do, show metabolites with a relationship to the metabolic pathway of choice. We use genomic annotation data, gene annotation data, to set up and make the foundation of our network of choice. Many high-throughput experiments, like microarrays, like RNA-seq, proteomics, metabolomics, things like that, those help us refine and add to our pathway. Okay, so for the flipped class, you're essentially going to be doing something like this. Each team is a group of collaborators. There is a bunch of metabolites. You'll be working on the same network, but you'll be working on it from different directions. So each of you in the group will individually get your own set of data that you will apply to your network here, your network that you're going to construct. First, you'll all be given the genomic data. They'll tell you the metabolic pathway of choice from substrate to product. Then you'll each report your findings to the team and construct your network and let the network take shape. And then maybe ask, or answer, I should say, some questions about that network. So that's what's going on, and I will see you tomorrow.